My name is Milan Stolpman, and today I'm going to be giving my presentation, which is called Soil Bacterial Communities Across Tropical Secondary Forests at Different Soil Orders and Successional Stages. So I am in the Dr. Maureen Spiota Lab of Biogeochemistry, and I work with my mentor, Emily Diaz-Vallejo, in the Department of Geography with UW-Madison. So to start off with a little bit of background information, soil bacterial communities are involved in a variety of really important ecosystem processes such as decomposition, symbiosis, as well as nitrogen fixation. And they're really important for cycling elements, and they influence global biogeochemical cycles. And so as we've had deforestation due to anthropogenic causes throughout the tropics particularly, and then these deforested areas have then been abandoned and allowed to regrow, the process of forest succession happens. And this gives us the opportunity to study the relationship between forest succession or forest regrowth, in other terms, as well as the successional patterns in the microbial communities that inhabit the soils. And so this investigation seeks to study exactly that, sort of looking at the unstudied or largely unstudied successional patterns between various bacterial phyla along the lines or variables of forest age or successional stage, as well as soil order. And so there are two main research questions here. One being, how do bacterial communities change across tropical secondary forest succession and soil order? And two, how does nutrient availability influence bacterial community structure and composition? So for the sake of time, I won't be able to fully get into this, but here are just some pictures. Here is the pasture, here is the young forest, and here is the medium forest, which are the three different successional stages that we used. And then here is one of the types of soil, sort of going back to that previously referenced term, soil order. Here's the alpha sol soil. And then here is the mollisol soil, which has, typically higher organic content. So here are the results. And so on this upper left-hand side here, you can see that this is an ordination plot, which essentially is getting at this message that there is clustering of soil bacterial communities across forest successional stages, as well as soil order. And so successional stage is represented through these different colors here. And you can see that there is clustering in between those different colors, with orange being on the left side, blue in the middle, and then purple on the right. And then you could also say the same for the shapes, which represent soil order. Next, and sort of the main message that I'm going to focus on is this graph here. And this is essentially showing that there are really variable responses in between the phyla. And so while proteobacteria on this side and actinobacteria really did not exhibit statistically significant relationships, acetobacteria here in the middle did show statistically significant relationships between successional stage as well, successional stage in terms of there were greater abundances of the acetobacteria phylum on the medium soils or forests, which you can see here. And so lastly, for our conclusion, so like I said earlier, there's a really variable response among various phyla across forage age and soil order, with acetobacteria exhibiting the strongest correlation. And yeah, there are just a lot of properties that go into this, such as pH, organic matter, and nutrient availability. And lastly, for future research,